on your bracelet, guys.
I will have to tell you sometime the story of Eric's and my trip to and the drive back in an unexpected spot. It was unexciting.
You remember, I guess, when Baltimore got those two back-to-back snowstorms referred to as snowpocalypse? And like nothing was plowed, and then Marnie and James Walter hiked to the Carnegie to feed the fish. I'm like, Marnie, they'll be fine for the day. She's like, no, we gotta go. Oh, and just suited up and basically rolled down the street.
Yes, I remember that well. I really, yes.
I don't.
Hi. So it is my great pleasure to have my friend Mary Goll come and give a seminar today at the GSI. And Mary and I was trying to remember how many years we've known each other, but I think it's got to be close to 20. Wow. Sorry. And so Mary and I actually did our postdocs in the same lab, but not at the same time. So I had just left Marnie Halpern's lab at the Carnegie when Mary started as a postdoc. And but Marnie has a habit of like making all of her trainees into one big happy family, and so Mary and I have had many chances over the years to interact at meetings, and I was trying to think of some embarrassing stories to tell about Mary this morning, but actually there were too many. I couldn't pick my favorite, so I told her I'd try to be a little well behaved at the introduction today. So Mary, as you can figure out by looking at the slide, works on zebrafish and studies chromatin regulation. And she did her undergraduate at Cornell and was a graduate student at Columbia in Tim Bester's lab. Is that right? Yes, where she first started working on methylation. And she continued that at the Carnegie as a postdoctoral fellow. And she was, if I remember this correctly, actually a joint postdoctoral fellow between Marnie Halpern's lab and Alan Spradling's lab working in Drosophila. But she saw the light and has been sticking to zebrafish work ever since. And she started off her independent career at Memorial Sloan Kettering. But I think for her peace of mind has and greatly enhanced life quality has moved to the University of Georgia a couple of years ago, and she is very happy there. She'll tell you all about it. And I've also noticed that I don't keep up with all the latest things because it's not exactly my field, but she's published several very impressive papers in just the last 2 years since she moved. And so with that, I will let you take over and tell us about your science.
Thanks, Shannon. Okay, I'm going to use the mic even though it's a small room because my voice is starting to go. Hopefully we'll make it through. I also definitely don't have any embarrassing stories to tell about Shannon. So I'll just get started with the presentation. So my lab is deeply interested in chromatin regulation in both the context of development and disease. And just to make sure we're all on the same page, DNA is wrapped around histones within the nucleus to form nucleosomes. And when we talk about chromatin, what we're talking about is the aggregate behavior of these nucleosomes. So understanding chromatin regulation is really important because disruption of chromatin is associated with abnormal development and a number of disease states. For instance, you'll almost always see abnormal chromatin contacts in cancer, in aging cells, and in a number of genetic disorders, including this one, ICF syndrome, which I'll talk to you about in the second part of my talk. So if you reach back in your memory to sort of your genetics textbooks, you may remember that eukaryotic genomes are segregated into two types of chromatin. There's open euchromatin, which tends to be the transcriptionally active regions of the genome, and more condensed heterochromatin, which is packaged more tightly, it's transcriptionally silenced, and it's important for genome stability. And you can actually see the difference between these two types of chromatin on an electron micrograph, where this dense heterochromatin is seen as these electron-dense blobs within this nucleus here, and the euchromatin sort of fills in the spaces in between. So despite the fact that we've appreciated this distinction between heterochromatin and euchromatin for nearly a century now, we still don't have a fundamental understanding of what drives the early fractionation of genomes into euchromatic and heterochromatic compartments during development. So this is one of the questions that my lab is broadly interested in. And when we think about this question, we approach it through the lens of constitutive heterochromatin. So for those of you who don't get to think about constitutive heterochromatin all the time, it's marked by this histone modification, histone 3 lysine 9 di or trimethylation, depending on the organism, and for us it's going to be mostly tri. It's enriched at highly repetitive sequences, including transposons, pericentromeric repeats around the centromere, some gene clusters, and telomeres. And it's important for a number of processes, including constraining transposable elements, regulation of these gene clusters, promoting centromere integrity, and suppression of recombination between repeats. So in my lab, we work with zebrafish because it's just the best. And over the course of the next 45 minutes, I hope I'll show you how my lab is really exploiting the advantages of the zebrafish system to uncover some new things about chromatin and heterochromatin establishment that might be hard to dissect in other systems. So I'm going to tell you two stories. The first one is kind of a mechanistic story about heterochromatin establishment in the early embryo. And the second one's going to be more about how we can use the zebrafish to understand the consequences of chromatin disruption in disease. So if you don't like one, hang on. Hopefully you'll like the other one. Maybe some of you will like both. Okay, so the first story starts with a postdoc in my lab, Catherine Lau. And she has some help from a really talented graduate student, Varsha Radshekar, who recently graduated from the Cornell program in New York through my lab, and Abigail Courtney, a talented graduate student in the lab next door to mine at UGA. And when Kat started out, she was just interested in thinking about the developmental regulation of heterochromatin establishment. So when we started this project, it had long been appreciated that in mouse, the histone modification H3K9 trimethyl, which is this marker of constitutive heterochromatin, is absent from the male pronucleus in the zygote and sort of accumulates there in subsequent cell divisions. It's also been known for a long time that in Drosophila, the early embryo lacks H3K9 trimethyl, at least at pericentromeric repeats by IF, and this has now been seen globally. More recently, it's been shown that this sort of developmental regulation of heterochromatin also occurs in C. elegans with increasing levels of H3K9 through early development. But at the time we started this project, we really knew about these two. We were wondering if this represented a general trend where there would be a lack of heterochromatin in early embryogenesis, or whether this was sort of two weird, unique phenomena that happened. So as all of you probably know, zebrafish is a really excellent model for understanding early development. We have external fertilization, means we get access to many thousands of embryos from the one cell stage onward, and these embryos progress through a stereotypical stages of development that we can precisely stage both based on morphology and hours post-fertilization, or HPF. And this first story is going to be more or less complete by the time we get to the mid-gastrula period around shield stage, or 6 hours post-fertilization. So when Kat set out, she simply said, well, what does H3K9 trimethyl look like in the early zebrafish embryo? And to do this, she initially did Western blots on bulk histones. And what she found was that there was essentially no H3K9 trimethyl, this canonical marker of constitutive heterochromatin, in early embryos until roughly sphere stage here. This is despite the fact that we could readily detect histone H3, so the histones are there. And I want to point out to you that this is more than 10 cell divisions the embryo is going through without this canonical mark. To sort of further confirm this, we looked by immunofluorescence, and again, in early embryos up until about the 1,000 cell stage, or 3 hours post-fertilization, we really see no H3K9 trimethyl in nuclei by immunofluorescence. At 3.7 hours post-fertilization, we can perhaps see the first signs of diffuse nuclear accumulation. And then by 4.5 and 6 hours post-fertilization, we see clear nuclear accumulation with these dense foci called chromocenters, which are that constitutive heterochromatin we see in nuclei. And this is what you would typically see in any somatic cell in any organism that you would look at. We also took a look by chromatin immunoprecipitation, either directed at the satellite sequences or ChIP-seq across the genome. What you can see is that there's a clear timeline of accumulation with little to no H3K9 trimethyl in the early embryo, and then accumulation by 6 hours at the places where you'd expect it. This is transposons, the pericentromere, other repetitive sequences. So if there are aficionados in the audience, you're probably aware that there's really two kinds of heterochromatin. There's constitutive heterochromatin, which is marked by H3K9 trimethyl, and there's facultative heterochromatin, which is typically marked by H3K27 trimethyl. This is polycomediated heterochromatin, and it's more commonly found in the context of gene regulation. But there have been contexts where these two can switch off with each other, and so we wondered if H3K27 trimethyl might be compensating for H3K9 trimethyl in this system. Turns out this is not the case. If we look at H3K27 trimethyl, we also see that this is limited in the early embryo with accumulation after the 1,000 cell stage. And this was actually first shown by Nadine Bastineau using chip arrays back in 2010. So this begs the question then, if we're lacking all of the canonical markers of heterochromatin in the early embryo, what does the chromatin look like at these stages? And to address this question, we turn to transmission electron microscopy. So just to show you that we know what we're doing, here's an EM of a nuclei from an embryo at 90% epivally, so this is just a typical somatic cell. And it looks like you would see in any textbook or in that earlier image that I showed you. So there's electron-dense regions around the nuclear periphery, as well as within the interior of the cell. Now check out what you see if you look at the 512 cell stage, when there's no H3K9 trimethyl or H3K27 trimethyl. This nucleus is completely devoid of condensed chromatin ultrastructure. People talk about ES cells being in a naive state, but it's not like this. This is just totally naive. We can follow chromatin across this window of development, doing EMs at a high resolution time series, and you can see again at 2.75 HPF, there's really no condensed chromatin ultrastructure within these nuclei. By 3 hours, maybe we're seeing some tiny little puncta here. These are really hard to quantify. If anyone here is an expert in EM quantification and can help us, I'd love to hear. By 4.5, we see these larger aggregates, which are more typical of condensed chromatin that you would see in a somatic cell. And these continue to progress through 9 hours. Now, these larger aggregates we've been able to quantify and using maximum thresholding. And you can see that they accumulate in this window between 3.7 and 6 hours post-fertilization. So on the one hand, when you look at that time series, you can say that that's a gradual accumulation of heterochromatin. On the other hand, I want to emphasize that this is actually really a period of roughly 3 hours in which you're doing this sort of genome-wide global reorganization of chromatin on a macro scale, which is really kind of profoundly amazing to me. So naturally, we want to use this system now to understand what are the mechanisms that drive this major reorganization and the global de novo establishment of heterochromatin during embryogenesis. And we don't have all the answers to this problem yet, but we do have one part of the puzzle, which I'm really excited to share with you today. So for those of you in the audience that work on zebrafish or have friends that work on zebrafish, it may not have escaped your attention that this timing of heterochromatin establishment correlates roughly with a really interesting transition in zebrafish development. And this is the maternal to zygotic transition. So this is the time in zebrafish development. It happens in all animals, although the time varies from animal to animal. When maternal transcripts, which have driven development up until this point are degraded, and we first have activation of zygotic genes within the genome. So naturally, we wondered if this transition was important for this, and we started to address this question by asking whether activation of the zygotic genome was required for heterochromatin establishment. So to do this, we injected embryos at the 1-cell stage with alpha-amanitin, which is an RNA pol-2 inhibitor, and then we assayed H3K9 trimethyl at 4.5 or 6 hours post-fertilization, times when we would expect this modification to be there. And you can see that there's a dramatic reduction in H3K9 trimethyl at both of these time points. Moreover, if we look by EM, you can see that all of this condensed chromatin ultrastructure is absent in the amanitin-injected embryos at 4.5 hours post-fertilization, suggesting that zygotic genome activation is important. Now, the simplest model for what would be going on here is that the H3K9 methyltransferases themselves need to be turned on at zygotic genome activation, and that's the bottleneck or the hurdle. However, this doesn't appear to be the case, because if we look in RNA-seq data, we find that mRNA encoding all of the known H3K9 methyltransferases, and SUVAR39, H1A, and H1B are probably the most relevant players in the system. They're all maternally loaded pre-ZGA at levels that are, if anything, higher than they are post-ZGA. So this suggests that those methyltransferases are there, and something is preventing their activity. Consistent with that model, if we take SUVAR39H1 protein that we know is active in a test tube, and we inject that into embryos at the 1-cell stage, we find that that is insufficient to promote earlier heterochromatin establishment, although we probably get a little bump in H3K9 at the time when we would normally expect it to be there. So this got us thinking about inhibitor models, and got us thinking about the possibility that perhaps there is a maternally loaded transcript that was preventing H3K9 establishment and condensed chromatin ultrastructure until after the maternal to zygotic transition. To address this, we started thinking about this really interesting microRNA, miR430. This is one of the first genes that's turned on at zygotic genome activation, and it is required for the degradation of many of the maternal transcripts during this transition. Now, when we blocked miR430 using a morpholino, or we used a morpholino targeting dicer, which is required for processing of miR430, what we found was that there was, again, a significant reduction in H3K9 methylation, supporting this model in which miR430 is required to degrade an inhibitor. So what's the inhibitor? We thought that was going to be a really hard problem, but we got a little bit lucky. So Antonio Heraldez, when he was a postdoc in Alex Shear's lab a while ago now, he had done the hard work of identifying miR430 targets at MZT. But I won't take you through this rigorous scheme that he put together to identify these targets, but I will say that if you look at the combined information from these, right at the top of the list of the best candidates was something that really jumped out to us. And that was SMARCA2, which if you work in Drosophila, you might know better as Brahma. And this protein is the catalytic subunit of a really important protein complex called the BAF complex, which has been implicated both in nucleosome remodeling and transcriptional activation, two activities that you might expect for something that was antagonizing heterochromatin during early embryogenesis. So we went in and we confirmed that SMARCA2 is indeed a miR430 target. SMARCA2 is downregulated at the maternal zygotic transition under wild-type conditions. And if we inject morpholinos targeting miR430 or dicer, we see sustained expression of SMARCA2 past the maternal to zygotic transition. So consistent with our model, we find that injection of a miR430 resistant version of SMARCA2 inhibits H3K9 establishment. We see reduced H3K9 trimethyl by Western blot when we inject this mRNA. But this is a tricky experiment, right? Because any kind of developmental delay will give us the same results, but not for legitimate reasons. So we were more excited when we saw that the converse was also true. So we find that depletion of SMARCA2 using either of two independently designed morpholinos actually leads to an acceleration in the timeline of H3K9 trimethyl establishment. So here we see the histomodification H3K9 trimethyl at 3.5 hours post-fertilization, a time when we don't have this modification yet in our control embryos, which was really exciting to us. So when we looked at where it was going, for the most part, it seems like this precocious accumulation of H3K9 is right in the spots that you'd expect it to be, which is interesting. It's at the pericentromeric repeats, and it's at the subset of transposons that would get H3K9 trimethyl a little bit later on. Now, some of you may not like morpholinos. We can have a beer and I can tell you why you're wrong later, but just in case, I can also hopefully convince you by showing you that chemical inhibition of SMARCA2 also accelerates the timeline of H3K9 incorporation, and it does so in a dose-dependent manner. So when we inject PFI3, which displaces SMARCA2 from chromatin, we see H3K9 trimethyl at time points when we don't see it in DMSO mock injected embryos. And the more PFI we inject, the more robust our signal is. So we wondered whether this H3K9 trimethyl incorporation that we're using as a segregate for chromatin was actually enough to facilitate the precocious establishment of heterochromatin at the ultrastructure level. And for this, we went back to our EMs. Oops. So what I hope you can see here is that when we inject PFI3 into embryos to inhibit SMARCA2, we also see an early initiation of chromatin compaction at the ultrastructure level in these embryos, suggesting that that precocious H3K9 incorporation is actually sufficient to allow for formation of, let's say, heterochromatin-like aggregates. Now, if you're an aficionado, you'll tell me that these aren't perfect heterochromatic aggregates. They're smaller and more dispersed, and I would agree with you. But nonetheless, it's enough to get this process going. This also suggests that there are additional factors working at this time that are working to establish sort of a more endogenous, like heterochromatic state. So the model here is that the maternal zygotic transition controls the timing of heterochromatin establishment through miR430 mediated degradation of SMARCA2. So at ZGA, we need ZGA to turn on miR430. miR430 then removes SMARCA2, and the removal of SMARCA2 is enough to facilitate H3K9 trimethyl and condensed ultrastructure following this transition. We're really intrigued by this model because it sets up a way to assure that at the time of zygotic genome activation, the chromatin landscape is fully open and naive, and that this heterochromatic or repressive regions are then built around that initial landscape of transcription factors that comes on at ZGA. And so now we're very interested to go back and ask whether accelerating the timeline of heterochromatin establishment influences the ability of these transcription factors to interact with the genome at ZGA. So some of you may be wondering what the mechanism is by which SMARCA2 is antagonizing H3K9, and we don't know yet, although we do have some ideas. So we're really intrigued by observations from Jerry Krabke in recent years, and what Jerry has shown is that BAF complexes containing SMARCA4, which is an alternative catalytic subunit, actually antagonize polycomb. They directly evict PRC1 from chromatin to antagonize H3K27 establishment or facultative heterochromatin establishment in somatic cells. And so we're intrigued by the idea that BAF complexes containing SMARCA2 will do something similar with constitutive heterochromatin, and that BAF complexes may actually have a more generalized role in antagonizing repressive chromatin states, with the catalytic subunit determining the specificity for the different kinds of chromatin. So for this first talk, what I hope I've convinced you of is that this initial fractionation of the zebrafish genome into euchromatin and heterochromatin is regulated by the maternal zygotic transition, that chromatin of the early embryo exists in this extremely decondensed or naive state prior to this transition, and that the chromatin remodeling protein SMARCA2 protects the early embryo against global heterochromatinization. So that's the first story that I have to tell you in a nutshell. And now I'm going to really switch gears, and I'm going to tell you a second story, or a second that focuses on a second focus in our lab. And that's on really asking whether we can use the zebrafish model to gain new insights into the roles for heterochromatin in human disease. And this is a story that was really motivated by a fantastic graduate student in the lab, Varsha Rajshekar, who's now doing a postdoc with Gary Karpin. So a tradition from Alan Spradling's lab is that when students and postdocs leave, they get a cake that has their project on it. And we've continued that in my lab. So this is Varsha's thesis cake, and this is Varsha's thesis project here. So if you're super astute, you don't need to listen to the second part of the talk because you can get it all from this cake right here. You guys have it? All right, and Varsha was helped out by a really talented UGA student, Alicia Higgs, who's picked up this project and is now pushing it further. Okay, so this is a story about 5-methylcytosine. So 5-methylcytosine, as many of you may know, is the addition of a methyl group to the 5 position of the cytosine ring in DNA. It's associated with transcriptional repression. It's found at more than 70% of CpG dinucleotides in vertebrate genomes, and it's absolutely required for viability in vertebrate species. However, a challenge is that while the importance of 5-methylcytosine on a global level is clear, the relative importance of this modification at particular subclasses of sequences has been difficult to define. And that's because really all of the tools that we've had to date get rid of it everywhere or nowhere, right? So we know that if you take it out everywhere, all sorts of bad things happen. But is that because of removal at one type of sequence or another? And Varsha was really interested in understanding the function of DNA methylation at a particular subclass of repeats called the pericentromeric repeats. So the pericentromeres are these highly repetitive subclasses of sequences that are found near centromeres in most animal species. And the sequence of these repeats varies significantly between species. However, they're usually there, and they're thought to contribute to centromere stability. Now, Varsha was interested in this subclass of repeats in particular because loss of DNA methylation at pericentromeric repeats is observed in basically all human cancers that you'll take a look at. It's seen in aging, and it's seen in a rare genetic disease called immunodeficiency centromeric instability and facial anomaly syndrome. Now, Varsha was interested in the implications of hypomethylation in these contexts, but these are super complicated. So she thought that she would focus on this genetic disease here. Now, the defining hallmark of this disease is a selective loss of DNA methylation at the pericentromeres. So here on the right, I'm showing you a control chromosome from a wild-type human stained with an antibody for 5-methylcytosine. And you can see that there's 5-methylcytosine all along the chromosome with particular enrichment here at the pericentromeres. Now take a look at this ICF chromosome here on the right. You can see that there's still this strong enrichment of 5-methylcytosine across the chromosome, whereas there's a significant depletion specifically here at the pericentromeres. And this has since been confirmed by whole genome bisulfite sequencing. Now, in addition to this really interesting molecular phenotype, patients with ICF syndrome show small stature, facial dysmorphism, some cognitive impairment, an overall failure to thrive, and a clear immunoglobulin deficiency characterized by hypo-IgM and variable loss of other immunoglobulins. Now, the reasons that loss of methylation, specifically at pericentromeres, would lead to these phenotypes are obviously not particularly clear. So, advances in understanding both the mechanisms that drive methylation at these repeats and the reasons for the phenotypes associated with this loss have been hampered by a lack of tools. So we know the genetic basis of ICF syndrome. This is a fairly rare human disease. It's a recessive disease, but it can be caused by homozygous mutations in any of 4 genes. So the first is DNMT3B. This is a de novo methyltransferase. In this case, there are compound heterozygotes for hypomorphic mutations. And that maybe makes sense if you don't think about it too hard, although if you think about it harder, you'll wonder where the specificity is coming in. The other 3 genes are 2 poorly characterized genes, ZBTB24 and CBCA7, and HELS, which you may know as LSH, which is a chromatin remodeling factor. Now, a challenge has been that when any of these genes have been mutated in mice, either through nulls or patient-specific mutations, what's been observed is that the mice are embryonic or perinatal lethal, and they seem to show more widespread methylation loss than is observed in ICF syndrome. So why mouse is not a good model for ICF syndrome, I can't tell you, but it's not. So Varsha wondered if perhaps fish might be a better model. And to tackle this, she mutated ZBTB24. So she chose ZBTB24 because at the time, these 2 genes had yet to be identified in ICF syndrome, and there are actually 6 DNMT3 orthologs in fish, and I fully support her decision to avoid that. Okay, so this happened, we started this project a little while ago, BEC or B-CRISPR, if you will. And so we made a deletion in ZBTB24 using talons rather than CRISPR, but it achieves the same result. And Varsha was able to get 2 mutant alleles of ZBTB24. The first is a large 7.9 kb deletion that takes out most of the protein coding sequence, ZBTB24. And the second is a 7 base pair indel deletion in an upstream BTB domain that leads to an early truncation. Now in our hands, both of these alleles behave exactly the same, and we've done all of the experiments with both of them, but I'll spare you seeing all of the data twice, and I'll simply present the data for the deletion mutant here. So the first thing that Varsha saw when she took a look at these fish is that they were viable, unlike the mice. But as adults, they were smaller than their wild-type siblings, both by weight and standard length. In addition, she was encouraged because they recapitulated other key features of ICF syndrome. So they showed a shortened lifespan. They showed facial anomalies. Now, I'm certainly not going to try to convince Shannon in particular that this facial anomaly is similar to the facial anomalies in ICF syndrome, but there are quantifiable differences in this fish face compared to this fish face. But perhaps most convincingly, we saw the same ICF-like immunodeficiency, where we saw reduced levels of IgM, IgD, and IgZ transcripts. Of course, we're not a human disease lab, we're a chromatin lab, and so what we really cared about was whether or not we would see the same loss of methylation at pericentromeres. And we were really excited to see that we did. So we found that when we looked at adults, there was an extensive depletion of 5-methylcytosine at pericentromeres. So for students in the audience who may not be familiar, this is a Southern blot, something that us old folks used to do all the time, but it's still really the best way for quantitatively looking at changes in repetitive sequences. So what we do is we take DNA, genomic DNA, we digest it with a restriction enzyme that will only digest the DNA if it's unmethylated, so it can't cut if it's methylated. We run it on a gel, transfer to a membrane, and then probe with our sequence of interest, in this case the satellite DNA. And what you can see is that while most of the DNA in wild-type fish is stuck up here at the top, or meaning it's methylated and resistant to digestion, when we look in 3 different adult individuals, you can see that there's substantially more digestion of the DNA. Now, people like sequencing and analysis of sequence, so we've gone in and we've looked at bisulfite converted DNA, and we've tried to count the number of reads that are converted, suggesting unmethylated DNA, versus not converted, suggesting methylated. It's tricky because of polymorphisms and things, and so I think that this is really a rough estimate of what's going on. But if you do this, you can see the same thing, that there is an increase in the unmethylated population of fragments in the ZBTB24 mutants compared to wild type when we specifically look at satellites, defined satellite sequences. Now, if we look across the rest of the genome, We were excited to see when we did enhanced reduced representation by sulfite sequencing that across the rest of the genome, DNA methylation appears to be pretty well intact. So we don't see significant differences in these violin plots of methylation levels at promoters, exons, transposons, or rDNA clusters. So at this point, I hope I've convinced you that ZBTB24 mutant zebrafish recapitulate the phenotypic hallmarks of ICF syndrome, including smaller stature, facial dysmorphism, immunoglobulin deficiency, reduced lifespan, pericentromeric hypomethylation in the context of an intact global methylation landscape. And this is exciting because this is the first faithful model of ICF syndrome. It's also the first viable animal model in which we have selective loss of DNA methylation at pericentromeres, which opens up a number of avenues of investigation. So before I keep going, telling you what we've learned so far, I'm going to pause and tell you how this story almost wasn't. And that's because when Varsha initially made the mutants in ZBTB24, she looked at 1 week post-fertilization, which is the end of the period at which rational zebrafish folks look at zebrafish. After that, we just throw them away. They're irrelevant. And at 1 week post-fertilization, you could see that the ZBTB24 mutant larvae were phenotypically indistinguishable from wild type. Moreover, if you did a methylation-sensitive southern blot, there really wasn't much difference. Now, Varsha went into a committee meeting and tried to tell her committee that this was a little bit more smudgy than this, and everybody kind of smiled in that encouraging but not encouraging way, and said, I don't know, maybe you need a new project. But Varsha said, no, ICF syndrome isn't a disease of embryos, it's a disease of juveniles, and I'm going to look a little bit later. And you already know the positive outcome of that story, because when Varsha looked in adults, she saw a clear phenotype. And what she found when she went back and looked carefully was that there was a clear sort of accumulation of phenotypes, and it correlated, here we're looking at length, and it correlated well with, here's the lengths, here's methylation. So we could see that starting at maybe 2 weeks post-fertilization, there was a small but significant loss of methylation at the pericentromeres, and this accumulated over time and correlated well with the onset of phenotypes in these larvae. So this really suggests that ICF syndrome is potentially a progressive disease, and that the role of ZBTB24 is important for sort of the long-term potentiation of methylation at these repeats. So it's performing like a maintenance function that is required over the long term. And this is consistent with some recent human data that's come out. So for the most part, ICF patients have arrived in the clinic late when they're experiencing phenotypes associated with the immunodeficiency. But there was recently a patient that was observed at 2 years old because of a sibling that already had the disease, and there seems to be a strong accumulation of phenotypes in that individual. Okay, so the other thing that Varsha realized when she looked at this was that this gave us a really unique opportunity to ask, what are the first consequences of pericentromeric methylation loss? There may be a whole bunch of secondary and downstream things that happen as that methylation loss gets severe, but what's the first thing that happens when you start to lose methylation, specifically at these repeats? And to do this, she turned to RNA-seq just to get a handle on what might be going on. And she chose a 2-week time point because at 2 weeks, these larvae are still phenotypically indistinguishable from their wild-type siblings. However, they do have a subtle but significant loss of DNA methylation at these repeats. So just like in any RNA-seq experiment, you can see that there are genes that are upregulated and genes that are downregulated. We couldn't make much sense of these downregulated genes here, but a really interesting signature emerged when we looked at the upregulated genes. And it confused us a little bit in the beginning. So what we saw was that methylation loss in ZBTB24 mutants was associated with activation of an innate immune response, or genes that were associated with response to virus. So this included interferon-stimulated genes, regulators of the interferon pathway, and inflammatory cytokines. So that was a little bit puzzling. So for those of you that don't think about the innate immune system all the time, I'll remind you that the innate immune system serves as a first line of defense against pathogen infection. So it's basically a lot of molecules that are capable of recognizing unique patterns associated with pathogens and eliciting an inflammatory response. Now, the other thing to take away from this slide is that this is complicated, and as you can imagine, well outside our expertise as a lab. So we thought that we would just try to get a handle on what was going on by asking what part of this pathway was relevant. And to do this, we took advantage of a feature of the innate immune system in that there are many diverse pathogen molecules that recognize different pathogens, but most of them signal through a few distinct adapter molecules. So the MAVS pathway is signals for different types of double-stranded RNA. MyD88 is response to diverse pathogen-associated molecules, mostly that signal through toll-like receptors. And the CGAS STING pathway recognizes unmethylated DNA in the cytosol. And you can bet that we had a bet on one of these when we were looking at this. Turns out it's a good thing we didn't bet a lot because we were wrong. So we mutated each of these using CRISPR-Cas9, and we asked whether mutation in any of these could mitigate that innate immune response we were seeing in our mutants. So when we looked at double mutants of the CGAS STING pathway, or MyD88, we saw that there was no change in the interferon response in these mutants, looking at two key genes that were upregulated within our data set, STAT1B and IRS7. In contrast, when we looked at MAVS, we saw that there was a significant reduction in both of these genes in the ZBTB24 MAVS double mutants compared to ZBTB24 alone. Confirming this finding, we mutated an upstream receptor, MDA5, which is typically involved in recognizing double-stranded RNAs. And we looked by RNA-seq, and you can see that this innate immune response signature is fully mitigated in these double mutants. So what's going on here? Well, I told you in the beginning that DNA methylation is typically associated with repression of transcription. And I told you that there was a significant loss of DNA methylation at the satellite repeats. So we wondered whether loss of ZBTB24 might be causing de-repression of a subset of RNAs that would be immunoreactive and recognized by MDA5 MAVS, and whether perhaps those RNAs might be coming from these demethylated satellites. So to test this, we first confirmed that indeed satellite transcripts are upregulated in ZBTB24 mutants. We've done this by northern blot. These are individual animals here. And we could see that this upregulation occurred during roughly the same timeline as the methylation loss that we were observing in these animals. But that's just a correlation. We also were able to show that both sense and antisense transcripts were produced from these satellite regions. I'm not going to go through this assay because it's a little bit convoluted, but first to say that sense and antisense transcripts were there, potentially allowing for the production of double-stranded RNAs, the known target of the MDA5 pathway. So finally what Varsha did was she asked whether these transcripts would be sufficient to trigger an innate immune response in wild-type embryos. And to do this, she took RNA for sense and antisense satellites, and that was in vitro transcribed, and she injected this into one-cell stage embryos. And she could see that at a relatively low dose, she was able to elicit an innate immune response as characterized by these 4 genes down here in wild-type embryos. What was really interesting to us is that she could elicit this response with satellite RNAs at a concentration where she couldn't get this response with just any old mock RNAs that she injected into the embryo, suggesting that it's not just the presence of double-stranded RNAs, but there are potentially actually immunoreactive motifs within the satellite RNAs that are triggering this response. And this potentially makes sense if you think about it a little bit. Throughout the genome, when you have RNAs that are transcribed regularly, they're going to be under some selective pressure to avoid having immunoreactive nucleic acid signatures within them. However, these satellite RNAs aren't normally expressed at all. Therefore, they're not under those same constraints, and they can evolve different variations in their nucleic acid signature that may be recognized by pathogen pattern receptor molecules if they were to be expressed. So the model then is that loss of methylation at satellite repeats triggers expression of satellite RNAs, which have a unique immunoreactive potential. These are recognized by MDA5, which signals through MAVs, triggering this innate immune response. So I hope today I've shown you that ZBTB24 mutant zebrafish represent the first faithful animal model of ICF syndrome and of pericentromeric methylation loss. And so we're now excited to go back and ask about the relevance of this pericentromeric methylation loss in other contexts, such as cancer, and we're currently crossing these mutations onto different cancer susceptibility backgrounds. I've also shown you that mutation of ZBTB24 is associated with selective and progressive loss of methylation of the pericentromeric repeats, that this loss of methylation results in de-repression of satellite-associated transcripts, and this finding really suggests potential roles for satellite hypomethylation in triggering autoimmunity in other disease contexts, such as senescence, where you have loss of methylation at these repeats. So with that, I'd like to thank my group in particular. This work was driven by Catherine Lau and Varsha Rajshekar. This is the rest of the folks in my group. They're super talented. These two are my kids. These are my collaborators. And funding, you can follow us on the internets if you like. Thanks.